thank you very much to the organizers for inviting me here. I know Hans-Peter for a long time, and we've been discussing on in vivo targeting for a long time, so let's hope it will happen soon in human at some point. First of all, for the community summary slide, I'm going to talk about 3 different subjects. Gene modification of progenitor T cells. Why is this a key finding? Because our key finding is that we can transduce them very efficiently. It might lead to long-term persistence of these cells. And how can it be related to cure? Because it would allow long-term gene therapy in T cells. The second subject will be on a in vivo T cell gene therapy. We, together with Christian Buchholz, developed new T cell targeted lentiviral vectors. Why is it important? Because the accessibility to, for gene therapy to more patients would be evident in that site, and the costs would be reduced to an enormous amount. And how is this related to cure? Well, I will show you an anti-cancer therapy, CAR therapy, but one can think also, of course, of HIV. The third thing is gene editing in HSCs, but in a safe way, and we developed nanoblades for the efficient gene editing. On-site deletion and correction would be the important issue here, and specific deletion of genes like in HIV in a safe way would be the outcome. And I think at the end of the talk, you will understand why all this is exciting. Okay, so first, what about if we do a stem cell transplantation? I think this audience all knows that we have an innate immunity recovery in a couple of weeks, but the problem is, and somebody mentioned this yesterday, that the T cell recovery is much, much slower and can take up to 1 year. That's why we came up with the idea to do the same is true for gene therapy, to try to in vitro generate these T cells, these progenitor T cells, and correct them in vitro, and co-infuse them in the end with hematopoietic stem cells. So which are the targets we would like to go for? These are the early thymic progenitors and progenitor T cells, which would be the ideal candidates since they are really primed towards the T cell lineage. And together with Marina Cavazzana from the Imogen Institute, the Necker Hospital in Paris, we applied a culture where we cultivate a CD34 on a delta 4 ligand, which is normally expressed by the thymus, by the human thymus. And this pushes the differentiation, as you can see here, towards progenitor T cells. Now I have to talk 2 minutes about a novel pseudotype that we developed, which has at its surface a baboon envelope, a retroviral endogenous envelope from a baboon. And what we did is we modified this envelope to be nicely incorporated on the surface of a lentiviral vector. The specificity of the envelopes are amino acid transporters, ASCT1 and 2. And what we've shown is that they efficiently transduce resting T cells as resting B cells as also hematopoietic stem cells, for example. So our question was, what would they bring in these in vitro generated progenitor T cells? And as you can see here, VSVG, and it's known for thymocytes, that they are very poorly transduced by VSVG pseudotypes. We have low transduction efficiency, while these baboon pseudotypes transduce very, very efficient. So we have a tool to modify these progenitor T cells. And what we did is compare actually the transduction of CD34 directly, injection into an NSG model, and on the other hand, the generation of transduced progenitor T cells and injection into the same mouse model. And what we saw is that we have, of course, a much quicker reconstitution of the T cell lineage, as you can see here, compared to CD34. And this is, for example, a graph showing the difference between the two. So I cannot go into detail, but we used in vitro this vector to correct gamma C deficiency in an XKIT patient CD34 cells successfully. And we showed also here that we have a much faster development of mature T cells compared to the corrected CD34. So this is a really nice approach, which one could also apply to HIV protection of CD4 cells, for example. The second part, I'm going to talk about a CAR T-cell therapy. You all know the successful stories about CAR T against the CD19 molecule on, for example, chronic lymphocyte leukemia and other B-cell lymphomas or leukemias. What we wanted to do is to try to in vivo generate these CAR T cells. But for that reason, our lentiviral vector need to be targeted specifically, of course, to the T cells. The vectors need to encode for the GAR-T, and the advantages will be that we have no ex vivo culture process, the cell phenotype will be conserved, and we will have a much lower cost of the therapy, of course. And it will be, for that reason, more accessible to patients. So together with Christian Buchol's lab, we developed a strategy where we inserted single-chain antibodies against a specific receptor on the T cells. In this case, we chose CD8 in the Nipah virus envelope, as you can see here at the surface of a lentiviral vector. And what we saw is that these, first of all, why is this so easy to do is because the Nipah virus has the G protein that recognizes the receptor on the one hand, and the F protein, which allows the fusion. So when you inhibit here the wild type receptors recognized and change the tropism, you don't touch the fusion protein, which permits still a very nice entry into the cells. And as you can see here, these Nipah retargeted vectors exclusively transduce the CD8 cells, while, for example, a VSV-G vector will transduce all the blood cells, as you can see. And the mutated Nipah doesn't transduce any of these cells. So this shows a very high specificity. I have to mention that this system is enormously flexible, so you can retarget it to fairly any receptor that you would like. And what we did for our CAR therapy, we developed the CD8 retargeted vector, encoding for a CAR against CD19, and we humanized mice with CD34 cells. We primed them 2 times with interleukin-7 to activate a little bit the T cells in vivo, and then we injected the CD8 retargeted vector. And as you can see here, we get a fair expression of the CD19 CAR exclusively on the CD8 cells in vivo, as well in the blood spleen as in the bone marrow. But what was more remarkable is that coinciding with this, we had a disappearance of the CD19 cells in the blood, in the spleen, and in the bone marrow to a lesser extent, though. So this means that this is a therapy that really could work in vivo. Of course, we do agree that we don't have a functional immune system here, and we need to switch to macaque to make sure that there are no adverse events here. So just to summarize this part, we injected in humanized mice the CAR encoding CD8s, which exclusively transduce RCD8 cells, the CAR T in contact with the B cells will make an amplification of these cells in vivo. That's why you don't need to hit so many cells in vivo. And it will make disappear your human B cells in the model. The third part of my talk is going on a new editing system that we developed, which my colleague called nanoblades, for an obvious reason, because they're small blades in a way. And the way he developed it, so Philippe Mongeau and Emiliano Ricci, was by actually fusing the Cas9 to the GAG protein. And what we actually do, we express two guide RNAs. I will come back to the envelopes in a moment, but basically it's an empty virus-like particle with no genome information. The Cas9 is incorporated as a protein, and the guides as an RNA. So they just are used as a shuttle to get into the cell of interest. The way we produce them is fairly simple. You transfect all the plasmids encoding for all the components into 2,9,3-T producer cells, and you have the nanoblades released from the cells in the supernatant. Now, there we come again to the tropism. We wanted to retarget them to our cells of interest. And as I told you before, this particular envelope is interesting for targeting T cells or efficient transduction of T cells and hematopoietic stem cells. Even in the case of hematopoietic stem cells, when you do not stimulate them, which is interesting because then you don't induce differentiation, of course, and they don't lose their stemness. So as you can see here, we mildly stimulated IL-7 with IL-7 T cells for 3 days. And when we applied the nanoblades, we had, after 24 hours, a very nice gene editing strategy. Now, with a single guide, probably this is much higher still in efficacy, but we still need to test this. But at least with 2 guides at the same time, which is easier to detect by PCR because you cut out a big chunk of DNA, you can see that we achieve already 30% in the T cells. Now, this was not on the program, but after discussion with Hans-Peter, I still wanted to give you another tool that we developed. And what we actually developed was a vector, again pseudotyped with our baboon envelope, but encoding for an HCV neutralizing antibody, so hepatitis C neutralizing antibody. And we developed a system in which we could force these mature B cells that are transduced to force them into plasma cell differentiation by co-injecting human CD4 cells in an immunodeficient mice model. And in 4 weeks' time, we have our plasma cells. Now, the strategy that we used here is a bit different from what was previously shown. What we actually have made is a vector that will express in the mature state on the B cells the membrane-anchored antibody, but when it would encounter anti-HIV, it will force differentiation into plasma cells and release antibodies in the supernatant. So this process is really driven, the construct is made in this way, that it is a natural way of secretion driven by the differentiation. So I would like to acknowledge all the people in the lab in Lyon, and for those who don't know, I also work in Nice in another lab on cancer and metabolism. Thank you very much for your attention.
Time for a couple questions. Yes, sir? The first VSVG pseudotype virus that you're using in vitro, is that replication competent or incompetent?
The baboon?
Yeah, no, where you were doing the gene editing, where you were actually used the VSVG vector, the pseudotype virus. That was the first one, because you had one later on that obviously was just a shell.
No. Actually, what we do is there's no targeting involved there. Of course, the message here is that we can combine all these strategies and we can retarget our nanoblades by putting a CD4 targeting envelope. In this case, we co-pseudotyped the nanoblades with VSVG and the baboon. And the reason was, empirically, we showed that in that case, we entered easier, on the one hand, easier into the target cells, right. But the VSVG was apparently necessary to have enough Cas9 into the nanoblades. It helped to increase the cargo.
Okay. Thank you.
Thank you.
So, can you incorporate donor DNA into your nanoblade?
Sorry?
Can you incorporate donor DNA into your nanoblade? So
no. The only thing that we've shown in the paper that's already published is that you can short donors, like oligos, you can stick them at the surface of the nanoblades. And like a flag or something short, that works fairly well. But these huge chunks of DNA, it's not easily packaged into the nanoblade, no.
Thank you.
At least we have not tried that.
Sorry, just a very small technical question. In those nanoblades, do you still need to express the viral protease?
No, actually, it's true that the Cas9 is linked by a protease site to the GAG, but the protease is in the cell. Once it's released in the cell, it's a cellular protease that will cut and release the Cas9.
So the particle's fine without the viral protease?
Yes.
Great. Thank you very much.
Thank you.